You say this.
Okay, let's get started. I hope my voice will last through today. We'll give it a try. My goal is I've still got some people that are making up the exam. My goal is to have it done for you by noon tomorrow. I will send out an email as soon as it is ready, and they will be down in the admin's office when they're ready. So just watch your email, but probably tomorrow about noon. Any announcements for class, Dr. Morton?
You know what time?
You got time? Okay. I'd like to start out just as a little review by showing you just a little short YouTube clip. This isn't the greatest animation of protein synthesis, but it is functional. One of the things that you might notice here is that when they talk about the ribosome, they only talk about two sites. They talk about the P and the A site. They do not talk about the E site. That's going to vary from book to book. Some textbooks don't consider the E site or the exit site actually a binding site, and so just be aware of that particular discrepancy. So let's take a quick look here. Hopefully the sound will come
The
through.
information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has two binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again.
So there's just elongation cycles now going on.
This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
Okay, I did a little checking around. I said last time that I thought that there was a mistake somewhere. As it turns out, whoever made this slide appears to have made a mistake here. This isn't 2 GTP, this is actually 1 GTP. So the information that I gave you previously, which kind of counted up for the energy demands for protein synthesis, was correct. So approximately about 3 ATP/GTP are consumed for every amino acid that is incorporated in that polypeptide chain. One of them is involved in charging the amino acid to the appropriate tRNA. One of them is consumed by binding of the incoming tRNA to the A site. That's where the mistake is in that one illustration. And then one tRNA is required for translocation. So again, for a small polypeptide, about 120 amino acids in length, that's going to chew up about 360 ATP. And if you ballpark it at about 30 ATP per glucose, that's going to give you 12 glucose molecules that are going to be consumed to make just that small polypeptide chain. So energetically, pretty demanding process. Okay, when polypeptide chains are synthesized in the cell, many times it's important for a lot of translation to occur in a very short amount of time, particularly if there are instances where that polypeptide chain is used for something that's in kind of high demand. So what can happen is that you can have kind of simultaneous translation of one message by several ribosomes at a time. They kind of line up behind one another, kind of like trains on a track. And then as one ribosome moves down the polypeptide, or the messenger RNA, and makes a polypeptide, quickly following behind it is another ribosome and another ribosome. That particular situation is called polyribosomes, and this really increases the efficiency of protein production tremendously. So here is a ribosome that is almost finished translating the complete polypeptide chain. When that reaches the stop codon, it will then dissociate and release. The ribosomal subunits will separate, the polypeptide chain will release. But then here's another one that was following behind this ribosome, and it's almost complete. Here's another one that followed that. Here's another one that followed that. Here's some incoming ribosomal subunits just starting synthesis at the 5' end of this message. So this is referred to as polyribosomes. And you can actually see these in electron microscopy. So this is a cell, and this is the messenger RNA along here. You can't really see the polypeptide chains that are being formed, but each one of these little kind of globules right here represents ribosomes, all of which are lined up on that message, kind of simultaneously translating it. Now, one of the things we talked about early on in the course was some proteins are destined to stay in the cytoplasm of the cell. Other proteins are destined to be exported. We talked about what differentiated those. The ones that are going to remain in the cytoplasm of the cell were made on free ribosomes, and the ones that were going to be exported were made on membrane-bound ribosomes. But how does a messenger RNA know which kind of situation applies to it? Okay? Why do some messages associate with ribosomes that then attach to ER membranes? If that happens, then that polypeptide chain, as it's being translated, is going to kind of snake its way into the cavity of the endoplasmic reticulum, move through the ER, likely be packaged up into a vesicle, and then move to the plasma membrane, or maybe move to the Golgi where it's going to be further modified. Well, as it turns out, all messages began initial translation by free ribosomes, whether they're to be secreted or not secreted. So here we've got a ribosome with a message, and here's a little bit of the polypeptide sequence that's being synthesized as that message is being read. The key is that encoded into the genes for these messenger RNAs that are going to code for secreted proteins is a little sequence called a signal sequence. And that signal sequence basically says, protein, you are destined to be secreted. If you lack the signal sequence, then you're not going to be secreted. So what are the consequences of that? Well, if this represents a message that encodes for a secreted protein, then this portion of the polypeptide chain has already been synthesized during translation, and this little portion coded for by the signal sequence in the message produces a little short segment on the protein called the signal peptide. And once that signal peptide is synthesized, it can then bind to a protein called a signal recognition protein, SRP. Okay, so this protein has already been synthesized by protein synthesis, and it's hanging around there in the cytoplasm. So if it sees this signal peptide, it will bind to it. And now this particular complex can dock to a receptor protein in the membrane of the ER. And that's going to latch that ribosome down onto that ER membrane. So now, when the remainder of that protein is synthesized, that protein will start snaking its way into this particular channel, and eventually that protein will move through the channels of the ER, be packaged, put into a vesicle, be excreted from the cell, go to the Golgi, or be modified a little bit later. So really, the genetic information about where these proteins are going to go is encoded into the genes. Okay? And that genetic information is then translated into a little sequence that allows that protein to be secreted. These signal peptides are removed before protein synthesis is completed, so the final polypeptide chain lacks this little signal sequence. Okay? So that answers part of that question about how are proteins sorted intracellularly? How do they know where to go? Well, as it turns out, it's encoded into the gene for that particular protein. Most of what we've talked about so far has been discovered and worked out initially in prokaryotic cells, more than likely E. coli. There are a lot of similarities between transcription and translation in prokaryotic cells and eukaryotic cells, but there are some fundamental differences as well. Things are a little bit more complicated in terms of transcription and translation in eukaryotes than they are in prokaryotes. Particularly, during the process of transcription in eukaryotes, a molecule called pre-messenger RNA is the first molecule that's really transcribed. Sometimes this is also referred to as the primary transcript. This is yet not considered to be a functioning RNA molecule. So the primary transcript is not the same thing as the messenger RNA. Now, it is in prokaryotes, okay? The primary transcript in prokaryotes is the message. But in eukaryotes, this pre-messenger RNA, the first thing that's copied from DNA, has to be tweaked a little bit before it actually functions as a message. We're going to talk about 3 modifications that result in this being converted into this. Okay? What are those modifications? 1, there's a process called polyadenylation. I'll show you what that is in just a second. This is a modification that occurs at the 3' end of the pre-messenger RNA. Second modification happens at the other end of the message. It's called the 5' cap. And the weirdest one involves actual removal of some of the nucleotides from the pre-messenger RNA. We actually cut out segments of that message and then stitch together the remaining portions that are left. And that is our functional messenger RNA. Those segments that we cut out are called introns. So let's take each one of these and dig into them just a little bit more detail. Okay, poly-A tail. What is it? It's added to the 3' end of the message. And this happens after transcription. So all three of those are post-transcriptional modifications. Basically, the poly-A tail is a little sequence of just A, somewhere between 30 and 200 of those that are stuck on to the end of the message after it's been translated. What are the roles of this? Well, we're not absolutely sure, but there are some good hypotheses. And here are some of the roles of this poly-A tail that we think might apply. Number one, we think that it might have something to do with regulation of transport of the messenger RNA out of the nucleus. Because remember, all of these post-transcriptional modifications occur within the nucleus. And then, for translation to occur, that modified pre-mRNA, the final product, messenger RNA, has to leave the nucleus, go through those nuclear pores, and then get into the cytoplasm to associate with the ribosomes. So the thought is that this poly-A tail somehow regulates that transport process. Okay. Another thing that we're pretty certain that it does is it regulates the degradation of the message within the cytoplasm. Remember that I said that molecules don't last forever, that they tend to turn over. And that's actually an important part of regulation of gene expression. If once you made a message it always was there, then you would continually translate that message into a protein. What if you wanted to stop making the protein temporarily? The message would still be there. But if that message is constantly turned over, then that gives you a place where you can regulate gene expression. We know that the longer a message hangs around, the shorter that poly-A tail gets. Some people have referred to this as being kind of a molecular hourglass. You know, that old timekeeping device where the sand goes through. And as the message gets older and older, that poly-A tail gets shorter and shorter. And the shortening that tail likely is a signal to the cell, it's time to degrade this message. It's been around long enough. It's time to get rid of it. There's also some thought that the poly-A tail might help to position the messenger RNA onto the small ribosomal subunit during initiation. As we were walking in, Dr. Morton said, about how long has this stuff, been around? He said this, wasn't when I was an undergrad, definitely. And I could answer very definitively. Poly-A tail was discovered in 1978. And the reason that I'm so certain about this is that's when I started grad school. And as an undergrad, I had never had to give any kind of a speaking seminar. I've never spoken before a group before. That's one of the reasons why we like you to do a lot of that. It's a good experience for you. And all first-year grad students in my program had to give a seminar before the faculty and other graduate students on some new topic. And my advisor said, there's this thing I've been reading about called this poly-A tail. Find out about it and tell us all about it. I was scared to death. Never having had to give one of those before. It was supposed to be an hour long. All your professors are sitting there, watching you, waiting to grill you. And your fellow grad students are bored out of their minds, wanting this to be over. So it really is a good experience for you, so don't feel like we're torturing you. It really is the kind of thing you have to get used to doing. About the same time, we found out that there were modifications occurring at the 5' end of the message as well. And this is called the cap. It's a modified guanine nucleotide, and I'm not interested in talking about how it's modified, but there's a slightly tweaked modified guanine nucleotide that's stuck onto the 5' end of the messenger RNA. What's its role? Well, there seems to be some overlap between poly-A tail and 5' cap We think this helps to position the message onto the small ribosomal subunit as well. And we also think that this has some role in protecting the messenger RNA from degradation. Okay, so a couple of similar kind of themes there. But the most bizarre discovery about messenger RNA. Oh, okay. So here shows you kind of pre-mRNA being converted into messenger RNA by the addition of this poly A tail and by the addition of this 5' cap. Okay. But the most bizarre thing about messenger RNA that we've discovered is that many, probably most eukaryotic genes we've discovered now have nucleotide sequences encoded within them that do not code for amino acids. These non-coding sections of messages are called introns. The introns separate segments on the messenger RNA that code for amino acid sequence. The coding segments are called exons. So introns split up exons. Messenger RNAs that have these particular introns are coded for by what we call split genes. This was a very remarkable discovery, and it didn't make a lot of sense to people because these segments are present in the DNA. They are actually transcribed into a message. So the introns are still in the message.
What's the difference between a codon and an exon?
A codon is a segment of DNA that specifies amino acid sequence. So exons would consist of a bunch of codons. Okay? Good question. So DNA contains these segments, introns, which are copied into messenger RNA. But before the messenger RNA can be translated, those non-coding segments, these introns, have to be cut out. And then the exons have to be spliced together. Well, what's that all about? That seems to be really wasteful and also really dangerous. Why wasteful? Because transcription does take energy. So why would the DNA have segments within a gene that didn't specify amino acid sequence? Why would we copy those into a message and then turn around and cut them out? And this seems bizarre. And if they're cut out, they have to be cut out properly. What if you cut out, when you're removing introns, a little bit of an exon? Then that would alter that whole message. And that would alter the polypeptide chain. And that could result in change of function. That could result in mutation. So people reason to themselves, there must be some kind of a reason why these introns are there. Otherwise, natural selection would have favored against them. Okay? Natural selection tends to select for efficiency. And this is not efficient. And this has some negative consequences associated with it if it doesn't happen properly. There are rather remarkable little molecular machines that are somewhat analogous to a ribosome called a spliceosome. And these little molecular machines, as it turns out, are really very good at splicing out introns and removing them without making really nasty mistakes. So, here's a pre-messenger RNA. It's already had its cap added. You can't see down here, but it's probably already had its poly A tail added. And then it consists of two exons separated by an intron. Okay, this intron doesn't code for amino acid sequence. So this exon codes for part of the amino acid sequence of the polypeptide. This codes for the rest of it. So what we have to do is we have to be able to accurately cut this out and join those two exons together. So when the ribosome starts moving down the message now, it will go amino acid, amino acid, amino acid, amino acid, amino acid, okay? It'll make the contiguous entire polypeptide chain. So how do we cut these things out? Well, in the nucleus, because this is post-transcriptional modification, several proteins are involved. One set of proteins interact with a type of RNA called snRNA. You're thinking in your mind, messenger RNA, tRNA, ribosomal RNA. Haven't heard of that one yet. This is a new kind of RNA. So there are genes in your DNA that code for these snRNA. If you're interested, SN stands for small nuclear RNAs. These are really pretty small molecules. They are single RNA strands, but they fold up into three-dimensional structures like a tRNA does. And snRNA associates with several proteins to form this structure called a SNRP. Small nuclear ribonucleoprotein. Don't worry about that. SNRPs associate with other proteins and form this molecular machine that cuts out introns. So here's the spliceosome. Here's a SNRP. Here's a SNRP. Here's another one. Here's another one. And then there are all kinds of other proteins that make up this complex. SNRPs recognize the boundary between this exon and this intron, and they recognize the boundary between this exon and this intron. And they bend the message, basically. And this causes this segment right here to loop out. These have, excuse me, endonuclease activity. Remember that term, endonuclease? And actually, it is the RNA molecule that is the endonuclease. So it's a ribosome. And that results in these bonds being broken right here. The cutout intron is released. And then those exons are bounded together to form an intact messenger RNA. So now we're finally ready to translate this messenger RNA. This is the mature, final messenger RNA that's ready to hook up with ribosomes and be translated. This is a pre-messenger RNA that contains some of this unwanted, unnecessary information stuck in the middle of that gene. So, the big question, why would chromosomes carry around all that extra DNA that isn't used in the final message? It's wasteful. And if they didn't cut it out, those genes could not be properly translated into protein. You can't translate something that's got a bunch of junk in the middle. It's expensive to maintain these sequences. Cutting them out is a risky business. Given these disadvantages, there must be an advantage to having introns. So people debated about this for a long time before they came up with this hypothesis, which seems to be at least the currently accepted idea of the benefit of introns. We believe that introns are involved in the evolution of protein diversity. One of the things that really surprised people about the Human Genome Project. Do y'all know what the Human Genome Project was? It was actually a government-sponsored initiative to sequence the entire human genome. As it turned out, it was successful. It was highly successful. in part because the process evolved so it wasn't just government research, but actually an incredible partnership developed between this government-funded research and private industry. And the success of this program was really driven by advancements in computer technology and database management. Craig Venter, who kind of turned tables upside down and developed what he called a shotgun technique to sequencing, drove down the cost of this and the time that it took to sequence just incredibly. The Human Genome Project is one of the very few government-sponsored initiatives that has ever come in ahead of schedule, they beat their original timeline, and under cost. When has that ever happened with any kind of a project? It's unheard of. Okay? The cost of sequencing keeps being driven down and down and down, and it is driving all kinds of interesting research from developmental biology to environmental ecology to medicine. And being able to sequence genes has become kind of the basic tool that is driving a lot of molecular biology. One of the outcomes of the Human Genome Project that was really just baffled people was we originally thought that given the size of the human genome, that there was probably enough genetic information to encode about 100,000 genes. And people thought, well, that probably makes sense because that's a lot of different proteins that can specify cellular phenotype. But as the Human Genome Project continued, that number of human genes kept being revised down and down and down. And now we're thinking that there are less than 20,000 human genes. That really surprised people because that hardly seems enough to encode for all the protein diversity that's needed to make a complex organism, like a human cell, like a human brain cell, for example. Well, we think that introns helps to explain this, what seems like an inconsistency. When splicing of introns occurs, as it turns out, there is more than one way, if you have multiple introns and multiple exons, that splicing can occur. And this particular phenomena is called alternative splicing. So here's an example of how alternative splicing might occur. There's a gene involved in the production of a muscle protein called troponin. And troponin T, which is one of the troponin proteins, this is what its gene might look like. 5 exons. Excuse me. Separated by introns. Okay? So, this troponin T gene is transcribed into a primary RNA transcript, which contains both the introns and the exons. And then, eventually, these introns will be cut out. And you would assume that the way they would be cut out is this would be cut out, this, and then one would be joined to 2, 2 to 3, 3 to 4, 4 to 5. And that would make then a troponin messenger RNA that could be translated into a troponin protein. Okay? But as it turns out, when splicing occurs, some spliceosomes can look at this and go, I'm going to splice together 1, 2, 3, ignore 4, and then 5. Other spliceosomes will look at this and go, I'm going to hook together 1, 2, 4, and 5. So 3 is left out of this one, but it's included in this one. That is alternative splicing. Now, how do those spliceosomes know how to do that? Why do we have different spliceosomes? Those are issues that get a little bit beyond the scope of what we can cover in an introductory course. But basically, the same genetic information can be reshuffled by this alternative splicing to generate different messenger RNAs, which will then encode different proteins. So one gene actually contains potentially the genetic information to form multiple different types of polypeptide chains. And that's a relatively recent discovery and a very exciting discovery. So, here's our basic summary of transcription and translation. Kind of throw everything together now. Okay? So here's the nucleus. Here's the cytoplasm. Here's our DNA. In the process of transcription, one of those two strands of DNA, this coding strand down here, is being copied into a pre-messenger RNA. The pre-messenger RNA has the tail added. They don't show the cat being added over here. Introns being spliced out. And then this final messenger RNA goes out of the nucleus into the cytoplasm. The message associates with ribosomal subunits. tRNAs attached to the ribosome and the message reading the genetic code, one code word at a time. The ribosome moves down the message in the 5' to 3' direction, lengthening the polypeptide chain until a stop codon is reached. In the cytoplasm, tRNAs are constantly being used to pick up specific amino acids. And these charged tRNAs are what carry the amino acids to the ribosome. And the anticodon of the tRNA recognizes the correct codon. And this anticodon-codon pairing results in the correct amino acid being joined to the polypeptide chain. That is dictated by the sequence of nucleotides in the message, which is dictated by the sequence of nucleotides in the DNA. Hey, Jenna.
What happens if the stop codon is one that gets messed up?
We're going to talk about that. Okay? Like that's going to be really the next topic. Consequence could be that if the stop codon got messed up, then translation would continue. Another question might be is, what if a codon changed into a stop codon? Then the polypeptide chain would be terminated prematurely. It wouldn't finish being translated. So, that directly leads into the next topic, and that is mutation. For our definition, we're going to talk about mutation as an alteration in the nucleotide sequence of a DNA molecule. So there's a change in that ATGC sequence that make up the code words of a gene. Now, some mutations involve large segments of DNA. We may duplicate an entire chromosome. We may delete large sections of a chromosome or maybe an entire chromosome. We can chop up a DNA molecule, a chromosome, and then rejoin it incorrectly. That's called an inversion. Did you guys talk about these last semester at all?
Just a little bit.
Just a little bit. Okay. What's probably the most widely known chromosomal duplication that we know? Trisomy 21, okay, Down syndrome, okay? An extra chromosome, number 21, ends up inside the cell because of a non-disjunction effect. It's the failure of chromosomes to separate properly during mitosis and meiosis. Well, we're not going to talk about these large chromosomal mutations. Instead, we're going to talk about point mutations, which alter maybe one or at the very most a few nucleotides within a gene. These point mutations can occur on their own. Typically, these happen as a result of an error in DNA replication. Now, remember that your cells have proofreading mechanisms built into the replication machinery that normally can detect these mismatched base pairings and correct them before the DNA molecule is completely synthesized. But occasionally one or a few creep through. And therefore, the daughter DNA molecule does not have the exact same base sequence as its parental template does. So spontaneous mutations typically involve errors in replication. On the other hand, induced mutations are caused by environmental factors. There are two major types of environmental agents that can cause induced mutation. We've already talked about one of them. What have we already talked about? Huh? UV radiation, yes, the thymine dimers. There are also a number of chemicals that can cause alterations in nucleotide structure. Some of these chemicals actually look like DNA nucleotides and sometimes can insert themselves actually into the DNA molecule, basically adding a nucleotide to that sequence. And that can have some pretty tremendous consequences if that happens. Well, we tend to think of mutations as being bad, right? I mean, a mutant is something that's, horrible, something that's gone really bad. Well, there are three real consequences of mutation occurring. And one is that there's no effect. We call mutations that occur but have no effect silent mutations. And I can pretty much guarantee you all are carrying around silent mutations in your genome, either as a result of errors of replication or maybe because of some kind of environmental damage that's occurred. Why could a point mutation particularly not cause any kind of effect? Redundancy of the genetic code, good. Okay, because remember that third code word in the codon can be different and still code for the same amino acid? What if an amino acid was altered? What effect does that have on the polypeptide chain? Well, it depends on what the amino acid is and where it's located. Okay, if I've got an enzyme and the active site is here, and then the rest of the protein is kind of folded around that active site, and I have a mutation in the gene that changes one amino acid out here, but here's the active site. Is that going to have much of a consequence? Yeah, probably not. But what if the mutation is in one of the amino acids as part of the active site? Yeah, then we're talking about some potential change there, some potential damage. Sometimes mutations are harmful. If they result in loss of protein function, and that protein function is critical to the organism, then that can be a harmful, maybe even a lethal mutation. I briefly mentioned in another context, I can't remember when, cystic fibrosis. That's a genetic disease. It is a single point mutation that actually alters the shape of a chloride transport protein in the membrane of cells, just a little bit. But the consequences of that altered transport molecule manifest themselves in all of these bad physiological conditions, which ultimately will result in premature death of people who suffer from cystic fibrosis. Occasionally, however, mutations can be beneficial. And the mutant gene encodes a polypeptide that actually is better suited to its function than the wild type was. Now, that's unusual, but it does happen. In lab, I think we're doing it this next week. We're doing the video. you're going to see a video called Accidents of Creation, and I mentioned this before. And it talks about the role that mutation has played in evolution of ruminants and grasses. And it's a very fascinating story that talks about how very small changes can result in dramatic evolutionary effects. So, with spontaneous mutations, typically we're talking about an error in replication. Why aren't they corrected? They usually are, but nothing's perfect. What outcomes might they have? They might be silent. They could be positive. They could be negative. We're going to concentrate a little bit down here and talk about these two situations, missense mutations and nonsense mutations, in this context of changing as little as one letter of the genetic code, resulting in something bad happening. So, here's a wild-type messenger RNA with a series of codons. And this specifies 4 amino acids: methionine, lysine, phenylalanine, glycine. So that's what these code words specify right here. So, this is a mutant sequence, okay? We've got one change, a base pair substitution of a U instead of a C. This should be a C right here, okay? That's the wild type sequence. Got a C over here. Instead, we've got a U, okay? But because of the redundancy of the genetic code, this still codes for the wild type polypeptide: methionine, lysine, phenylalanine, glycine. What if the mutation occurred here instead? Then this particular codon would have been affected. This still codes for methionine, this still codes for lysine, this still codes for phenylalanine, but now this codes for serine rather than glycine. This message does not make sense the way it should. This is called a missense mutation, and it changes a single amino acid in the polypeptide chain. Down here, we've had a different mutation that's occurred, not in the first codon, but in this second codon that normally codes for lysine. We substitute a U for an A. Seems kind of inconsequential, but UAG says stop. So in this particular situation, we would make a single amino acid, and then the ribosome would translate that message and go, it's time to stop it. That's all I'm going to make. So when a mutation results in a codon being turned into a stop codon, we call that a nonsense mutation. We don't finish the polypeptide chain. So these are the three basic kind of potential negative outcomes these are negative outcomes. This is a no effect outcome of having a single point mutation, which is typically the result of an error in replication. Next time we'll talk a little bit about, and we will follow up with that in the video, talking about missense mutations and how a single amino acid substitution can have a tremendous effect in a disease you may be aware of called sickle cell anemia. We'll use sickle cell as kind of an illustrative example of the possible effects of missense mutation. Okay?
Especially with my tax return.
So to take my exam today, do I have to come to your office or come here?
Yeah, come to our office and then we'll go back to the lab.
All right.
Okay?
Thank you.
People say you know it is, but by that,
Yes, it is. So we will just, we'll just record and, you know, there's nothing special that you'll miss.
Okay.
Okay.
Thank you.
Here yesterday.
Where were you?
Got update.
Pardon me?
I got update.
Come to my office, okay? Look.